Presence of Antimicrobial Peptides in C. elegans during regeneration by Brandon Reid and Quentin, Maine, in collaboration with Jennifer Morrison and Professor Ann Alman. C. elegans is one of the many simple organisms that researchers use today. In its most recent years, researchers have found axonal regeneration capabilities within this tiny worm. However, what does that mean for us? For us, this is a priceless discovery. To begin, axonal regeneration, to be simply put, is the body's natural capability to regenerate axons. Axons are very important pieces of the nervous system in all species. They are long fibers that are used to transmit information via electrical impulses to different neurons, muscles, and glands. Humans are especially capable of regenerating their own nerves and have been for years. However, the time frame of this regeneration has undoubtedly been random. Of course, the severity of the wound has always been taken into consideration, but most cases don't turn out the way they had originally seemed. Research on the C. elegans worm may let researchers understand the basics of axonal regeneration, opening new doors into diseases such as Alzheimer's and unlocking new strategies to enhance the natural regeneration process in human beings. Our question did not revolve around the medical uses of C. elegans, however. We were curious as to how the worm can go through the regeneration process without dying. We had hypothesized that as C. elegans regenerate, the presence of antimicrobial peptides can be detected. To go about this process, we would need a few days. Our first day included obtaining multiple types of bacteria, some known to be resistant to immune response, and inoculate them on agar plates that we had prepared that day. The bacteria we used was E. coli, S. aureus, P. aeruginosa, and B. cereus. We had then taken 4 sterile discs and placed them onto the 4 different agar plates and coated them with different amounts of C. elegans tissue. To obtain the C. elegans tissue, we swabbed a plate of growing C. elegans with the inoculating loop. We then submerged the loop into 50 microliters of sterile water, and to conveniently dispose of the worms, we had dipped them in liquid nitrogen. After the dip, and everything was all thawed out, we spun the worms down to collect their tissues. Next, we had taken the spun down tissue and pipetted 5 microliters of said tissue onto 2 discs on every plate. We then put 10 microliters of tissue onto the other 2 discs on the plates. We then incubated the plates for a 24-hour to 48-hour period. We had done this in hopes of seeing zones of inhibition where the antimicrobials would fight off the bacteria. Unfortunately, we had to restart our project due to human error before the incubation period. This is why we have no results nor pictures to show at this time. However, we truly believe that we will see significant zones of inhibition on all our plates. This strong sense of judgment comes from the idea that there has to be some sort of defense during the regeneration process that had been given to the C. elegans after years of evolution. What we plan on doing after we get our data can go in a variety of ways. If our data goes the way we had expected, we plan on going back to PLU to confirm our results, and then going the next step by seeing if we get the same results with live worms, if that's possible. If our data does not go how we expected, and we do not see any zones of inhibition on any of our plates, we will try new bacteria to see if we can achieve that zone of inhibition and then further our experiment. Understanding these antimicrobial peptides could exponentially